Cell biopsies, 100 times more data. The future of cancer detection is here. So today we're going to talk with Mohan Uttawar, who is the CEO of One Cell Diagnostics. I should say the CEO and founder of One Cell Diagnostics. So Mohan, thanks for coming on the show. You're very welcome. How are you today?
Very well. Thank you for inviting me.
So can you just explain to us your journey so that we can understand what One Cell is in terms of your life trajectory?
Journey is long, five decades. Started in a very tiny village in the middle of Bunis in India, where I was born. In a farming community of a village, we didn't have electricity, didn't have TV, no phones, no nothing. But fortunately, I was able to transcend the physical borders, and have been settled here in Bay Area for the past 4 decades. Engineer by education. I started my career here at Intel and with HP. And then my entrepreneurial journey started in the tech world. But a dear friend suffered through cancer, and I met some folks at Stanford. And when I saw a pathologist looking through the microscope with a mechanical counter in hand counting, I didn't feel good. In a place like Stanford in the middle of Silicon Valley, where we are celebrating uh, computers beating the chess grandmaster, why are these pathologists using mechanical counters to counting the cells? And that's how my entrepreneur journey changed into medical technology. And I started a company to digitize pathology. And ever since, it's been my passion for the past 15 years or so, focusing on applying innovation in technology to improve human health.
That's a wonderful story. So explain to me then what digitizing pathology means to a layman who's not a pathologist, perhaps.
Um, it's about pattern recognition. It's about
Mm,
well, what happens in cancer. Uh, there is no good early detection, so it's accidental discovery. And when they suspect something, the first call is always radiologic scan, CT scan or MRI. And they find something they suspect. The final call is made by pathologist, where you do what is known as the tissue biopsy. Okay,
mm.
so you uh, go deeper, you do the surgical section, you take a piece of tissue, then you cut it, you fix it, you stain it, and you create a glass slide with the staining pattern. And they put it under the microscope and they look at the patterns of the dots, the colors, the dots, the size, the shape, everything. And then pathologists are trained to looking at those images to say, it's cancer, or no cancer, or the stage of the cancer, so on and so forth. And they generate a pathology report. Based on that pathology reports, oncologists will decide the treatment. And that used to be the case for the last several decades. Unfortunately, uh, one shoe doesn't fit all. Okay. And so that's when personalization of treatment and therapeutic recommendation came about. That field now has evolved into what we today call it as a precision oncology. The personalization of the treatment, basically depends on biomarkers. Biomarkers are specific signals. You have genomics, you have transcriptomics, you have proteomics. These are different fields of sciences that are focusing on different things. But cancer is also like an elephant and six blind men. Each one of those individual blind person, depending on where they touch the elephant, they will describe the elephant like that. Genomics tells you something. Proteomics tells you something which is a little bit different. Transcriptomics tells you something different. Pathology, radiology, unfortunately, they all tell you a little bit, but nobody had figured out how to piece the whole thing together.
So just before we go on, can you explain to the viewers what genomics, proteomics, and what was the third term you used? What third?
Transcriptomics.
What was?
transcriptomics.
Transcriptomics.
Yes,
I
yes.
mean, I think we need just for clarity, what do those terms mean?
These are all science. Cancer is a genetic disease. It's very difficult to find out why cancer happens. And there are two
Mm,
types of main categories in cancer. One of them is the genetic form, right? And that's about 11-12% of the time. And the other one is hereditary in nature. But most of the thing is called somatic. The environment, what you eat, what you breathe, your lifestyle, that creates sort of a system. And unfortunately our immune system fails to fight the cancer. Okay, everybody has cancer cells, but what happens is if you have stronger immune system, they fight, they kill, and the life goes on. It's only when you're not able to fight it, they become dominant and they start growing. Okay. And so the science of study of genes in the body, there are 30,000 genes. Now, the turn of the century, you have the Human Genome Project that uncovered a lot of
mm,
these new areas that we started. And say, some medicines work for someone, but it doesn't work for other. What's the difference between the two? Okay. In fact, there are interesting studies where two, brothers, in fact, the twins, one works, another doesn't work. That's because there is a genomic nomenclature. The genes are a little bit different and not hugely different,
mm.
just a
We're
small
talking
difference,
about subtle differences,
very subtle difference,
right?
0.1,
Right.
0.2% difference, right? So that's the
So
study of
that's
genomics.
that's what I was going to say. That's the study of genomics. So proteomic or transcriptomics
Yeah.
and proteomics. What's that?
Okay. So transcriptomics, so genes get converted into proteins, and ultimately proteins is what you see. You know,
Yeah.
Genomics is like your roadmap. You open the Google Maps and say, I'm going from here to point A to point B. What's my route going to be like, etc. Proteins is like, hey, here, there's an accident, there's a jam. Now we got to take a alternate path, right? It's here and now. That's what it is. Um, the transcriptomics, you may have heard RNA, right? mRNA. With COVID, everybody start, Google University, taught or this thing, right? About this thing. So some genes get converted into protein. That subset of genes, okay, that get converted into proteins, the study of that is transcriptomics. And that is primarily done through what is known as RNA.
You've explained the difference. You've explained your motivation rather, which was to actually develop personalized diagnostics almost, and to effectively bridge the gap between the radiologist who provides the imaging, the MRI scan, and the pathologist who looks down the microscope. But let's move on and talk about what cellular pathology is all about. Because the title of your company is One Cell. So let's talk about cytology. Where does cytology come into this discussion?
Yeah, good question. So historically, precision oncology was first generation was driven by tissue biopsy.
Mm,
Like I explained to you, you take the tissue piece and you apply genomics, and great companies like Foundation, sort of made their mark on that. Necessary but not sufficient. You see um, that only small percentage of the population, get benefited, and tissue is always an issue because to get the tissue biopsy is not easy. It's a surgical procedure. It's expensive.
mm.
It's strenuous, right? So as the science evolved, liquid biopsy became popular. That's the second generation. You take small blood sample, 10, 20 ml, and then you do the analysis on the blood sample, and that has been shown that it's equivalent to tissue biopsy. Okay, because
So
it
just to be clear, we're talking about getting a blood sample and looking at individual cells that may have escaped from a tumor elsewhere in the body, rather than actually sticking a needle or a knife into that part of the body. Yeah,
correct, correct, correct.
yeah,
And
yeah,
it's not the cells, the classic liquid biopsy, they take the DNA
yeah,
fragments, collect the
yeah,
DNA,
yeah,
from the dead cells, and then
yeah.
you characterize those DNA, you analyze them, and you say what genes are being expressed, in those fragments of DNA.
Right.
Okay. Now that was second generation. Now, unfortunately, that's a good biomarker, but it doesn't tell you what is the current state of cancer in the body. These
Mm,
DNA are coming out of the dead cells. Okay,
right.
they are not a true representation of what's going on in the body. So
You're
tissue is in.
effectively look you're effectively looking at the rubbish you're looking at someone's rubbish, basically seeing if there's any signs of malignancy.
That's right. That's right. You're absolutely right.
Mm,
You're looking at the DNA fragments coming out of the dead cells, right? So the third
mm,
generation, which is where we come in, is capturing those circulating live tumor cells.
yeah.
Now, a cell has got at least two order of a magnitude more information than a DNA fragment floating in the blood. Unfortunately, it was not very easy to capture. So
Mm,
CTC, circulating tumor cell, is not a new field. 10 years ago, this was very hot area. And number of
yeah.
companies, lot of funding went in. And what they were able to capture and count. Okay. But it was not very popular because the science had not evolved to the level where you could tell the doctors what to do or what not to do. So you had situation like your CT scan or PET scan would say, "Oh, clean, clear." So patients are happy because their PET scans are clean. And
Mm,
then the CTC technology would say, "Oh, but I found two cells." And the
yes.
doctor would say, "Okay, what do I do?"
Yeah.
Okay. So a happy patient now turns into unhappy, and doctor's not able to tell what to do. And so
Yeah.
fraternity said, look, don't tell me the information, don't give me the data. That is no use to me because I can't take any action. And
Right.
that's why
Right.
the first generation of CTC, that entire field, did not pan out because science hadn't
So
evolved.
why are you different? Why
Good
is this different?
point. Two main differences. Good question. First and foremost, count and capture is not that's not a very useful endpoint. We are different because we are able to capture that one in a billion cell with high sensitivity, with high
Mm,
specificity, with high purity, okay, and high simplicity. So if I take those three angles, we are 1,000 times better than the CTC 1.0. The second groundbreaking invention that we have as a company did is being able to isolate that captured cell. So we have now live single cell, not dead cells or fixed cells, but live single cell. And now the science has evolved into single cell genomics, single
mm.
cell transcriptomics, single cell proteomics, and we apply the data science techniques to take now from that single cell genomics, proteomics, transcriptomics, and combine it and analyze it and provide meaningful insight.
So with this greater level of insight into the patient's clinical status, which has ultimately been derived from effectively one cell in a 20 ml blood sample, previously the oncologist would have said, "Don't tell me because I can't do anything with it."
Yeah.
What's changed now that the oncologist says, "Thank you very much, I'm not going to change treatment as a result of this information you've given me, Mohan."
Yeah.
What's changed in that regard?
Good question. So when you say that those tumor cells that stem out of it, remember, a gram of tumor sheds about 1 million cancer cells in a given day.
All
But
right.
we have trillions of cells, so it becomes ones in a billion. But they
Yeah.
represent what's going on inside your body. And now we are saying, so for instance, if you're a lung cancer patient, and if you say my CTCs are expressing EGFR, the
Yeah.
doctors know what medicine will work.
Right.
If they
So
express BRAF,
depending on
correct,
the markers, then you can tailor
correct,
treatment. But
absolutely.
here's so you can get a more refined treatment with a more personalized treatment as a result of the information derived from your one cell. But can you ever say that the patient is cured?
That's a very good question. There's no such thing as cure in cancer.
No, you
It's remission.
cannot.
It's
It's
remission.
remission. Yeah.
And you know,
Yeah.
95% of the time, the people in remission, there's one biggest question mark. You know it's going to come back. You just don't
Yeah.
know when, from
Yeah.
where, and how bad
Yeah.
it would be. And look at the current methodology today. The
Yeah.
screening that happens with CT scan or PET or MRI, you can't detect the reoccurrence until the secondary tumor has grown to 9 to 10 millimeter in size.
Yes.
That's often too little, too late.
Yeah.
Our hope is that we are able to detect way, way early, and we
Mhm.
are able to present information to the oncologists to what
Mhm.
to do about it.
So,
Uh,
so this is what I'm interested in. So, you've got someone that you've done a blood test in, and they've received tailored specific medication for their kidney cancer. So, you've done a blood test that has successfully targeted treatment for kidney cancer. So, blood to an organ. That patient has undergone appropriate personalized treatment,
Yep,
and the scans now then show remission.
yep,
Cancer's gone away.
yep.
At what point do you then say, "Oops, there's a problem"?
Mhm.
Is there a threshold number of cells per 20 ml blood sample, or is it even the emergence of one cell that then triggers, a kind of a second round of treatment? What's the threshold
Yeah,
beyond which you have to act?
yeah. Good question. And I don't have a great answer right now. We have, through our studies, said minimum three CTCs, in a 10 ml blood sample is our cutoff.
Ah, right,
And
right,
it's not that CTCs means problem, right? It shows the problem, but then we characterize it, we analyze it, and we say, hey, are there any mutations in that?
right.
Okay, is there a problem? No, then you continue to be in a monitoring. Now, historically, a CT scan or a PET monitoring has an exposure to these things, right? Radiation
Yeah,
exposure, and so you
yeah,
couldn't do very frequently. But a
yeah,
simple blood draw, it doesn't bother you that much, right? Of course there's a
yeah,
cost factor to it, but you don't have to wait for a full year till the next
yeah,
year's PET or
yeah.
CT scan could come. You could do more frequent stuff, and that decision will be made by oncologists who, when we present, we found the CTCs, the numbers aren't that bad, there is no mutation, then you could say 6 months. Okay. If you find mutations, you could adjust the longitudinal monitoring depending on the stage of the cancer, the type of a person, and
Mhm.
whatever the situation might be.
Let's take a kind of a broader view, if you will, for the moment. We've discussed the specific technique and obviously in very, very shallow detail. But to what extent okay, my first question is, why has this not happened before? What were the key ingredients that needed to come together to actually then produce one-cell diagnostics?
Yeah. Very good question. I think, yeah, I almost feel like it's a perfect storm.
Mm,
The science hadn't evolved to the level where we will have the sensitivity to capture that one in a billion cell.
mm,
And more so, the science had not evolved where captured cell could be isolated without changing the characteristics of that cell. Okay,
mm.
so that's the evolution of science. Secondly, the technology hadn't evolved where we could go deeper inside a single cell. Okay, to be able to do the whole genome sequencing, the whole transcriptome, the proteins, and it's an overwhelming amount of data. If AI wasn't there to analyze the
Ah,
data,
you
to
said it.
convert that into a meaningful actionable stuff. Look, oncologists are super busy human beings. Okay, they barely get 20, 15, 20 minutes with a patient. And look at the amount of information that is thrown at them. They're almost like a captain in a cockpit flying the plane in a very turbulent weather.
Yeah,
Okay,
yeah.
they need everything at their fingertips. Today, they use their educational, experiential stuff, an intuitive way, by intuition, they make that decision. Our goal is to transform that decision from based on intuition to data driven decision. It's an
But you said the key
overwhelming.
word there, didn't you? You said a key word, AI. This would not have happened without AI. Is that what you're saying?
Nope, nope, nope. You're
Right.
absolutely right. And you know what AI does and why I'm excited about, okay, is the ability to forecast, ability to predict. Our goal, our mission is to turn oncology into predictive oncology.
Mm.
Okay. And that is possible only because of AI and ML. So, good science, when combined with good AI technique, that combination is going to provide us the ability to manage the journey of a cancer patient while they're in remission.
You've developed this magical tool that, as you say, gives oncologists the ability to use data to personalize treatment. How widespread is this technology? Is it available everywhere in the United States? Is it available internationally? Is it available where I am from in Australia?
Yeah. It's just the beginning of the journey.
Right.
It's available limited here in United States, where I am.
Yeah.
We've also launched this in India. We made it
Yeah.
available there because, they're with shorter path. Australia is absolutely on my radar. There are two key opinion leaders there. We're working with initial studies. Um,
Yeah.
but the path we have taken is initially to go after the pharma and biotech companies that are doing clinical trials, okay, where they can use this technology to select the right patients, deselect the wrong patients, monitor post therapeutic monitoring, and then analyzing, is it working? If it's working, why it's working? If it's not working, why it's not working? That's the stuff. Now, we
Mm.
chose that strategy primarily because the acceptance of new science and new technology is much easier in the pharma and biotech world. Okay.
Mm.
We also need to conduct a lot more clinical studies to get the regulatory approvals, the FDA clearances, and then follow that with the reimbursement strategy, which takes 3 to 5 years,
Mm.
and that's a long journey. While we are on that path, shortening the path to revenue by providing these services to accelerating the translation research in drug development is a right thing to do for the company initially, and that's why we're doing that in US and Europe, and Australia is definitely on the radar for us.
So we're running out of time. So what I would ask is, do you have one message of hope for any viewer who may be touched by cancer, either themselves or a loved one?
Absolutely. The future is precision oncology in cancer management. The future of precision oncology is biomarkers. From a simple blood draw, a true single cell multiomics biomarkers is going to help them predict the future. Okay, turning cancer into a predictive cancer oncology so that we can act on it before it is too late. It's an exciting time for the industry. The convergence of science and technology absolutely gives a hope, okay, that you can manage and you can fight the battle and win the battle by providing these kind of technologies will make cancer, I won't say curable, but will turn cancer into a manageable disease, into a chronic disease, just like the other diseases like the diabetes and others. There's no cure for it, but you could manage it. When I moved to Bay Area, you know, HIV, the three-letter word, meant how many more weeks, how many more months.
Mm.
Today, that question is not asked. People have learned how to manage and live comfortably. My my crystal ball is through this combination of AI and new science, cancer will become a chronic disease that we could manage comfortably. So there is a ray of hope, and companies like us are pushing the envelope of science and technology to make it happen. That's our mission.
Mohan Uttawar, CEO of One Cell AI, many thanks for your time today, and many thanks for giving us all hope. That's all for today, folks. My name's Dr. Ferghal Armstrong, and this has been MedHeads.